Okay, so we will have exam 3, same as before, it's open book. I already been put the 2 sample questions on the eCampus. So you go ahead to do a practice.
What was that?
And the key I will put there over the weekend. And also don't forget about at least to read through all the slides and the class materials. Just give an idea what we talk about. So let's see what we have some practice question here. I'll just go over these, okay? So this is the question we talk about is applied microbiology, which is for the safety. So you need to know what is US FDA, FSIS, and the US EPA. What is that? This actually EPA I haven't mentioned Environmental Protection Agency, but we definitely mentioned CDC, so you should know. Okay, you can replace with CDC. So this is Food Drug Administration. So what they're responsible for, you can say the foods other than poultry, meat, and processed egg. You can say about that, okay? What is FSIS? That is Food Safety Inspection Service. And what they're responsible for? We talk about that is for the poultry, meat, including red meat and the processed egg. Okay, what is CDC? Centers for Disease Control and the Prevention. Okay, that is for surveillance, basically. So they're using PFG methods. Okay, so that's the first question. You can find on the slides, very simple. Okay, this is the second question, related to biochemistry. Which of the following process generates a great amount of energy? Okay, you have fermentation, substrate level phosphorylation, anabolism, glycolysis, and anaerobic respiration. Now this one, you're going to go back to see your slides, but you also need to know, definitely is not anabolism. That's using energy. Is that right? We talk about this catabolism. Definitely is not a substrate level phosphorylation. Because that's just the way it generates energy. You can say like 1 ATP, but it's absolutely not should be for the calculation. Okay, how many ATP fermentation generated? Only 2 ATPs. How many ATP glycolysis generated? We also say 2 ATPs. Well, if it's a single closed cycle, usually we mean that. So it's not the answer. So how about anaerobic respiration? This is somehow between 2 to 38 ATPs. And this is, in one of these slides, I just want to show you where it is. So since it is a review, I just want to show you where it is. Let's go back to see the slides, slides 1. We mentioned about the, not this one, this is about chemistry we mentioned, so not this one. Sorry, it's sensory. This one should be covered. Not yet. This one, 3. The biochemistry section, we had one slide. Yes, that section. We had one slide go down, talk about here. It's 36 or less. Look at here, corresponding to anaerobic respiration. Okay, so that should be the places what you're looking for. And I always say this slide is important. Okay, so that's something we want to emphasize about that. Good, let's go to see the questions. to the next question. So let's go move up a little bit. So we have all these. Okay, then we move these. The next question, also about chemistry. The transferring of electrons and the hydrogen by a dehydrogenase to an organic molecule as a terminal electron acceptor is called. This is the one we mentioned about at the beginning of the class. If you have an electron is a donor, and also the electron is an acceptor, if both the donor is organic chemical, acceptor also is organic chemical, this is called fermentation. And a very easy way is pyruvate become lactic acid. Now, of course, there is an NAD reduced to NADH oxidized become NAD plus. So, the transfer electron hydrogen by hydrogenase to an organic molecule as terminal electron acceptor is called what? That's called fermentation. Exam only going to have 4, okay, A, B, C, D. This is just practice that you not have so many choices. So that's fermentation. Then the next question is simple. Which of the following is the best definition of fermentation? We just said the oxidation of carbohydrates with organic molecule serving as a final electron acceptor. Okay, so that's a very simple question. Which of the following used as an electron carrier by living organisms? Well, we mentioned NAD, nicotinic adenine dinucleotides, nicotinic adenine dinucleotides phosphate, fulvene adenine dinucleotides. Obviously, all of the choices. So these are the things I think it's very reasonable questions, which tells you the basic knowledge of the biochemistry. If you want to review by yourself, then you could go this section, go to the top. Look at here. This slide tells you definitely what is called aerobic respiration, what is called anaerobic respiration. It's right there. Very simple. Give you the example. So this slide is also, you can mark it using a square or using a star. That could be useful in the exams. Okay, so that's give you some of the hint how to do these questions. Okay, let's go ahead to see other questions. Then we move on to the big one. We're going to review it again. So let's look at question number 6. Maybe this go a little bit large. Okay, question number 6. Question number 6. This is the same thing, like we just mentioned. Which of the following can be used as an electron acceptor during anaerobic respiration? Well, we talked about what means anaerobic respiration. This means you have an electron donor is organic chemical, and the electron acceptor is inorganic chemical. But the inorganic chemical other than oxygen. Because if it's oxygen, that's called aerobic respiration. This is called anaerobic respiration. So what are the choices? Well, that's pretty straightforward. You have a nitrate, you have a sulfate, you have a carbon dioxide, and all the choices. Well, that's easy. Okay, that's very simple. And if you want to review the slides, where are the slides you should be looking for? And we can go back to see the slides. There will be the picture right here. This is aerobic respiration, showing you all these examples for the electron acceptor, what type of the bacteria corresponding to that. That's all the examples. Okay, so that slide you can also mark it, will be useful for answering questions. if it's related to that. Okay, now let's look at the next question. The hypothesis that proton-motive force-driven ATP synthesis is called chemosmosis. Where's the answer? The answer, did I put there, I think? That's called chemosmosis. That's a chemosmosis. We mentioned it briefly, is the electron going to be transferred, coupled with the dehydrogenase, that's hydrogen, which is proton, repel out of the cell membrane system, then it's going to be accumulated there, generate energy, we call it potential energy. It's called a proton-motive force. Since it's a motive force, it's going to do something. It will goes back and activate ATP synthesis, and then generate ATP. And at the same time, coupled with activate transport and the rotation of the flagella. So that's a very good example, which is called chemosmosis. That's a terminology for that. So you can highlight on the top. It's chemosmosis. That's a terminology could be tested. So this is what we're going to prepare for the exam, how to do the review. Okay, so this is very simple. On the top is chemosmosis. Chemosmosis. Okay. The next question is simple. Organisms that form lactate, ethanol, and carbon dioxide as end products of glucose fermentation of, what is that one? It's called hormone, or you can say it's a hormone, non-hormone, or hetero fermenter. Okay, you say non-hormone or hetero fermenter is fine. Because glucose fermentation, if you generate only acid, let's say you generate only lactic acids, we call this is a hormone fermentation. And if you generate other than lactic acids, like ethanol, lactate, carbon dioxide, this is called a non-hormone or hetero, just say. Both are fine. Well, when you go for AEM 341 major class, you need to answer is hetero. But here, it's okay, okay? The hormone or non-hormone is fine. Okay, this is going to be the big one. The next question. Calculate the ATP generated during the aerobic respiration. So I make it a little bit small. This is going to be the question where I hope everybody can do it, because that's a very basic question. So I will be just put the slides there. So you're going to do the calculation. So how to do that? You don't need to know all the chemicals. We're not going to test your chemical. Let's say we had a glucose. Okay, we go here, generate pyruvate. Then go here, TCA cycle. That's the only thing you know is enough. We don't skip all these other chemicals, because we don't talk about biochemistry. So how you do the calculation? How many ATP used here? That's 2 ATPs used. We call it an investment area. So how many ATP going to be here? Somehow here have an ATP generate. Somehow here has an ATP generate. And what else we have? Somehow here has ATP generate. Somehow here has ATP generate. But what the ATP generate comes from? This is from NAD reduced to NADH2, and this is NAD reduced to NADH2. Okay, so how many is it generate? This will be 3 ATP, that's 1. So you add up together how many? That's 8 ATPs. There's 1 here. So that's 5. Multiplied by 2, that's 8 ATPs. Okay, now let's go here to see. This transition step, acetyl-CoA. there is one NAD reduced to NADH2. There is one NAD reduced to NADH2 happening here. There's one NAD reduced to NADH2 happened here. There is one FAD reduced to FADH2 happened here. And the NAD reduced to NADH2 happened this here. And there is a one GDP generates GTP. So how many? 3 right here, 3 right here, 3 right here, 1 here, 2 here, and 3 here. So 1, 2, 3, 4. 3 ATPs multiplied by 4, add 1, add 1, 2. That's 15 ATPs. Since we have a 2 cycle, so 15 ATPs multiplied by 2 equals 30 ATPs. And these two add up together equals 38 ATPs. So that will be the calculation, pretty straightforward. Okay, that will be the pretty straightforward calculation. Sometimes in the exam, I want to make your life a little bit easy. I maybe just ask you to calculate this section. Now, my major course, I talk about AEM 341, I ask them to draw the chemicals and then calculate a little bit more detail, and then we give the extra credits if the students know all the chemical structures. We're spending like 30 minutes to talk about this, but here we just be briefly. If you don't know all these chemicals, but you know the places happened is ATP generate should be enough. So, related to this question, what are the slides you should look in for? The slides what you should look in for is basically this one. Is that right? This is the slides what you should look in for. This is absolutely the slides you should look in for. They're very important. And then you go down here, you should look in for this guy. Now, what's the difference between that? The only difference between these two is the top one this one here is NAD reduced to NADH2 is 2.5 ATPs, and FAD reduced to FADH2 it is 1.5 ATPs. The older one is using 3 and this is 2. That's the only difference. Okay, other than that, it's the same. So this is just something I want to let you know. You could use this one if you're comfortable with the biochemistry test, because you already learned in the other classes. If you're new, you never learned the biochemistry, I would say you can use this one, which is relatively easy for you. Basically easy for you. Okay? So that's something we want to mention about it. So let's see the next question. This is the one that's just confusing you. We have so many choices, but it's not difficult. If yeast metabolized one molecule of glucose to ethanol alcohol in the absence of oxygen, how many net molecules of ATP would be obtained? So this is a long sentence, but you don't mind to care about this. This whole thing tells you yeast in the absence of oxygen could do fermentation. Now, no matter how complicated it could be, fermentation is only 2 ATP going to generate. That's the answer. Except it's making a vinegar. That's a different story. If you have a wine, you open the bottle, you put it in the room temperature for extended period of time, have a secondary reaction, become acetic acids, that's a different story. If I have time during the presentation, we're all going to jump in, talk about vinegar manufacturing. But I just want to let you know, here we talk about the simple fermentation, does not mean how complicated, it's only 2 ATP going to generate. Okay? So the reason is making manufacturing of vinegar, that is a secondary fermentation is involved. So this is kind of straightforward. There's 2 ATP generated. Okay, next one. How aerobic respiration and anaerobic respiration are similar. That's a concept we already talked. So how they are similar? Let's look at these choices. Both use oxygen as a terminal electron acceptor. Of course not. Oxygen for aerobic respiration, not for anaerobic respiration. There are other names for glycolysis. Of course, this is wrong. Both utilize inorganic molecule as terminal electron acceptor. That is correct, because this is what the answer for. Neither receive electron from glycolysis is wrong. Anaerobic does not exist for microbes. Of course not. Lots of the anaerobic respiration bacteria is. So that's pretty obvious. Both use inorganic molecule. Okay, oxygen or others. Question 12. Probionic bacteria makes probionic gases. Acidic gases, carbon dioxide in Swiss cheese. So if you know about this, you should know this is this guy, this is this guy. That's acidic gases, probionic gases. What is this called? Not anaerobic respiration, not a substrate level phosphorylation, not the end product of repression. That's called fermentation. Very straightforward. Swiss cheese. You have this carbon dioxide in the hull, that's fermentation. Okay, it's straightforward and simple, but it's a good question because Swiss cheese, some of you eat every day. You see the hole there, it's carbon dioxide, it's probionic gases. And by the way, this is antimicrobials. This is short-chain fatty acids. This is also short-chain, very short-chain fatty acids. Those are lipids. We can talk another topic. In aerobic conditions, it takes sugar to produce the same amount of the ATP when compared to anaerobic conditions. Well, of course, it should take sugar to produce the same amount. It should be less. Is that right? Because aerobic respiration, you reacted with oxygen, you have a completely electron transport chain, you generate enough ATP. Anaerobic. So if you want to dig this one a little bit, so we talk about NAD, go FAD, go FeSCOQ, FMN, then we do cyto, chroma, CA3, oxygen. So we have this, we have this. So why? Do you know why? You look at the distance. This voltage compared to here is a longer distance. That means energy going to generate. If it's anaerobic respiration, they not do this electron transport chain. They do a modifier. Somehow start from here. So it's short. That's not much energy because the distance between the electron donor and the acceptor, the voltage value difference is much shorter. That's why anaerobic respiration is not enough ATP compared to aerobic. So that's a part of the reason to explain. So that's less is the answer. Okay. So that's question 13. Let's see whether do you have any other questions? Okay, no more. That's good. So this one we finished. We want to talk about another topic, which is genetics, because that's always lots of people will be confusing. Especially some of you have not taken science class yet, so we want to do all these questions. Let's look at these one by one. Two strands of DNA molecule, that is they are oriented in opposite direction. What is this called? Anti-parallel Is that right? They run in opposite direction. One is from 5 prime to 3 prime. Another is 3 prime to 5 prime. Anti-parallel. A consensus sequence in the untranslated leader sequence of prokaryotes RNA serve as a ribosome binding site. What is this called? What is that called? Shadagano sequence, is that right? AGG. Because ribosome binding needs to recognize that. Does not mean every single piece they could do. Amino acetylTRNA synthesis catalyzes covalent addition to what to the tRNA? This you're going to look at the slides. Just look at the slides. What are they going to bind it to? Just look at the slides. tRNA. Where are they binding to? Attached to tRNA, catalyze the amino acidyl tRNA synthesis. So amino acidyl tRNA synthesis. The answer is that. Covalent synthesis catalyzes addition to the tRNA. Let's look at here. Let me just take a look. That's right there. Attach amino acid to the tRNA. So the answer, you could add right amino acids. Okay? That's amino acids. Genetic information of DNA is divided into units called, that's easy, genotype. What it means genes, do you know? Genes means genetic, carrying genetic information of the DNA sequence. That's called a gene. Does not mean every sequence is gene. Okay, they only carry genetic information. The basic difference between DNA, RNA DNA residue in the sugar and the pyrimidine basis, RNA has ribose and uracil, whereas DNA has deoxyribose and thymine. That's true. That's correct. And we talk about these things are very interesting. If you know about this guy, you should know. What are the difference? Let's go back here, then go here. What are the difference? This is uracil, and this is thymine. What's the difference? We say the methyl group at carbon 6 at an unimportant position, replaced with proton. So that's uracil, that's thymine. Okay, only one difference, because originally is a methyl group, right now become a proton, so what happened, a hydrogen group, what happened, it's not stable. That's why uracil is not very stable. Okay. Which one is not a pyrimidine? So we have purine and pyrimidine. And the pyrimidine. A, T, G, C. So which one? Purine. That's one. It's not pyrimidine. That's pyrimidine, pyrimidine, and pyrimidine. That's purine. Is that right? Adenine is purine. And the adenine, guanine are purine, cytosine and thymine are pyrimidine. So which one is not? Adenine. So the answer is A. Okay, next question. Which of the following is not a complementary base pair usually found on the DNA? This is easy. This is everybody. Okay, this one. Move down a little bit. That's not a difficult question. This is everybody knows. In the DNA sequence, it's not a complementary in the DNA. A and T, G and C. This is A and U. This is our RNA. So that's the answer. Don't always choose that one, okay? You have to read it. Some of you don't even read it. Whenever they see D, all of the choices, they will just pick this. No. Does not mean all of them pick all of the choices. Okay, just let you know, you still have to read the question. Which of the following is true about the structure of DNA? Purine and the pyrimidine base attached to the one carbon of the deoxyribose sugar. This is correct. Purine and the pyrimidine base are joined by phosphodiester bond. Yeah, that is correct. Adjacent bases are stacked on top of each other. One base pair is 0.34 nanometers. So that's correct. That's all of the choices. Which of the following nitrogenous bases is usually found in RNA but not in DNA? Uracil. So that's easy. Next one. The DNA is replicated. The daughter molecules containing one strand and parent of DNA and one strand of newly synthesized DNA. This is called semi-conservative. We mentioned about that. We mentioned about that a couple of times. Is that right? 5' to 3', 3' to 5'. And remember, we're using the daughter bands will be M15. This is Matt Mendelson and Frank Starr research to mention about that, using the isotope of the nitrogen with Sicilian chloride to do the spin down and then to see the different precipitation. That's all you learn in your biology class. So that's semi-conservative. Okay, next one. Y shape of DNA molecule where the actual replication process occurs. Always remember this is called replication fork. That's very important. It's a place called a replication fork. During the during replication of two strands of DNA molecule are unwound from one another is called. This is, of course, is wrong. Nothing called unwitness. Never happened. Primaries, of course, is not. That's called primarization. Ligase, this is to connect Okazaki segments. Of course, the helices. Is that right? They will be unwinded with each other. That's not the unwindness like this or primaries. The coding sequence in the DNA of is normally continuous. That is, it's not interrupted by night coding area. This is the one. Test your wisdom a little bit. These are the same. Is that right? Called the eukaryotes. This is prokaryotes. This is eukaryotes. So, which one? Well, of course, it's A. Otherwise, you're going to pick all of that. And I don't have all of the choices. So that's A. This is one you know. There is a sequence right here, then here, then here. Then you have to cut. So they're called the intron. And they're called the exon. And the intron and the exon have been cut, then connected. So that's in genetics class you mentioned. But we skip that one here. Just to let you know, the prokaryotes DNA is continuously and there is not interrupted with non-coding sequence. Okay, that's called a, that's what this question is. Okay, let's see other things. The region at which the DNA wait a second, let me see. The region at which the RNA polymer is binding. What is called? This is a very simple question. Remember, we talked about this a little bit complicated. 5 prime to 3 prime, 3 prime to 5 prime, and the start place because of +1. Then we have a conservative sequence which TA, TA, AT, that is -10. Trigonal box. This is 16, 18 base pair difference. This is TTGACA. What is this whole thing called? A promoter. And this is the universal promoter. That's called -35 for all the bacteria. But when you're regarding their function, the promoter could be different. And the promoter recognized by sigma factor of the RNA polymers. Okay, so that's promoter. Next one. The unexpressed region of the split genes is called, that's intron. The exon has to be connected. That's intron. This question I haven't mentioned, to be honest. You can kick it out. Oh, I won't test you. Trigonal box, I just mentioned. What is trigonal box? Trigonal box is also called -10. So what is trigonal box? Approximately 10 base pair upstream. That's the sequence is TA, TA, AT. That's called a trigonal box. Okay, this comes out with the slides. It's very important. You should know. Let's go back to see the slides. Which one? Not this section. Let's see. The should be the exam 3, lecture 1. The slides. This is the slides is important, and you should mark it. Is this guy? This is important. This whole thing is promoter. That's a conservative sequence called a -10 sequence. This is -35. And the -10, we give them a terminology. We call this is a trigonal box. And I could also ask you to write down the sequence of a promoter, -10 and then -35, and then you should draw these. You should write down these. Okay, there's a bunch of box and there are a bunch of box. And then the whole thing right here is a promoter. Okay, so you should know. This slide is important. And I think this slide is described very well. Look at the promoter region. Look at what they said on the top, promoter region. That's really good. Okay, that's important. This is a slide you put a mark 3 star. That will be useful in the exam. Okay, so let's go talk about the other questions. The genetic code is said to be because more than one codon may specify for a particular amino acids. Of course, it's not called ambiguous. It's not called multiplication repetitiveness, degenerate. Is that right? So this said you very clearly is in the slides. You could show you. You don't have to remember. We don't test you that. Isoleucine, AUU, AUC, AUA. So isoleucine. Violin, GUU, GUC, GUA, GUG. Did you feel about that? The box number 3 is flexible because it's a wobble impact. Is that right? There or GU. That's called a wobble. That's called a wobbling. Can be flexible of the box number 3. So you can see it. It's except MET, methionine. It's AUG. Because it's start coded. Otherwise, it's all can be 2, 2, 4. That's called the degenerate. which means redundancy, but in the genetics textbook terminology, they don't call about that. Okay. Which is not a termination codon? What is a termination codon? AGG. Sorry. Which of the following is not a termination codon? This is, AUG is the start codon, so it's not. Molecules delivery amino acids to ribosome during translation. Which one? Transferring it. Okay, carry-on anticodon, but it's carry-on the amino acids. Let's see 20. The portion of the tRNA molecule that is bonding to the codon of tRNA is called, well, that's called anticodon, I just mentioned. You always remember this guy here, a tRNA, go here, go here, 5 prime, 3 prime. This is anticodon. That's called anticodon arm. Then you have a T theta C, a variable arm, T theta C for arm, different arm. Okay, that's anticodon. In ribosomes can attach to the mRNA and begin translation, even though transcription has not been complicated. Same thing. Prokaryotes, because of these eukaryotes. Even you don't know, you're going to pick A, because if you pick B, you're going to pick C. If you pick C, you're going to pick D, and there is no B, C, D. But sometimes the multiple questions could be tricky. Is that right? In the other classes you have, in your biochemistry class, it could be 2 of them above, 3 of them above, B or C, B, C, D. That could be like 7 choices. Don't let you guess. But here, it's a simple question. You don't even need to know, because prokaryotes, that's the only choice. So sometimes you need to be smart. It's very important in the exam, always like that. So that's a very straightforward question. Let's see the next one. The reaction sequence links adjacent amino acids to the growing polypeptide chain. That's called transpeptidation. You never heard about these. What is this? Of course, it's called transpeptidation. These questions, honestly, it's not difficult. What it means? It means nonsense confusion, which means you never heard about this terminology. You only heard about transpeptidation. So these obviously ACD is not the choice. Even you don't know, you still can pick the answer. So that's not a very difficult. Okay, now let's go see some of the basic question I want you to know, because it's important. Okay, since you're in here, we're going to talk about